this video we will discuss about tumor invasion mechanism in brief. So tumor invasions means that the ability of the tumor cells to penetrate in the surrounding tissue. So different kinds of tumors works in a different way, and this tumor has uh, sometimes also been related to these migration strategies. So in terms of the migration strategies, how the tumors are the tumor divides itself in the term of individual tumor cells can migrate, or the collective tumor cells as a group can migrate from its one part of the from one organ to another organ, or from one site of infection to another site of infection. So what it has been identified is that in the uh, amyloid kind of migration strategies has been observed in this kind of cancerous cells such as lymphomia, leukemia, SCLC. So this amyloid means that a group of cells migrates, changing the tissue, surrounding tissue, uh, and in the case of this sarcoma and geoblastoma and anaplastis tumor, what will happen is that the single cells move through these mesochemicals with the help of mesochemical characteristics. So what happens in this case is that the cells will release some kind of protease that will degrade the surrounding environment, or a surrounding extracellular matrix, and helps the cells to move from one place to another place. So these are about the single cells, and in this case, in the top two case, the function of integrin and protease are low compared to the bottom case. The bottom kind of case is the cluster and covert, and this kind of movement are observed in epithelial cancer cells. So in this case, the uh, cell-cell interactions plays a important role of the cells that are in the periphery, and at the same time, the categories and the integrins functions increases compared to the first two case, and this is the exaggerated version of cluster and covert, that is multicellular strands and sheets can move from one part to another part, and these are found in this epithelial cancer or vascular tumors. And there is also a possibility that there is a change in the migration strategy observed from one phase to another phase, and that is shown in this brief here. Is that in the case of this, when the multicellular strand sheets are migrating, so some of the cells might dissociate from this sheet, and since these cells does not have catherines, so they will transfer themselves from epithelial to mesochemical characteristics, and they will start to producing proteases that will degrade the surrounding environment and helps these cells to move from one to part to another part. And if they don't have these proteases, if the some mesochemical kind of cells does not have the proteases, because of the suppression of those proteases inhibitor due to the some drug injection, then they will change from mesochemicals to amyloid transition. So in this case, a group of cells will move, and this time they, instead of degrading the extracellular matrix, they will change the shape of the surrounding tissues or extracellular matrix, that will help them to move from one place to another place. And if this beta integrin is inhibited in this kind of transition, then it will shift towards, it might shift, the amyloid kind of transitions might shift towards this, oh no, not this amyloid, if from this case, yeah, from epithelials to, sometimes it may also shift towards this clusters kind of shape, and these things might happen. One of the reasons could be that amount of cells are less as compared to the multicellular strand sheets, and in this case, so this is usually occurs when the clusters separate itself. It is these things divided into two parts, and the last part is the transition is about when this cluster does not have the beta integrin, or beta integrin is inhibited because of the drug injection, then the transition occurs from collective to amyloid, uh, collective uh, tumor invasions to amyloid tumor invasions or collective to amyloid transitions occurs. So in this case, the cells will invade or migrate. Cell will invade and migrate via this amyloid mechanism. That is, they will try to change the shape of the surrounding tissue instead of degrading the surrounding tissue. So I hope you got it. And beta integrin is kind of a, a cell surface marker that helps in the uh, that helps in the this gap junction. So the disruption of this might leads to the shifting of the collective to amyloid transitions. So I hope you got a basic idea about how this tumor invasion mechanisms works. And if you need to understand more about this tumor invasion mechanism, you can go through the reference that has been provided in the description. That's all for this video. Thank you for your time.